This is the third portion of the genetics lessons. Last unit, we talked about sex chromosomes. Remember, females have two X chromosomes, and males in humans have an X and a Y chromosome. Remember, X chromosomes are much larger than our Y chromosomes. This means that X chromosomes carry more genes than a Y chromosome. When we look at genotypes of X-linked traits, this means that traits are found on the X chromosome only. We see that females can have homozygous genotypes, or they can have heterozygous genotypes. When writing these genotypes, we need to keep track of our sex chromosomes. So remember, females will always have two X's. So in this case, our female can be homozygous dominant, X big B, X big B, or she can be homozygous recessive, X little b, X little b. Notice that the traits themselves, or the alleles, are written as superscripts on the X chromosomes. Females can also have a heterozygous genotype, being X big B, X little b. Males, on the other hand, only carry one X chromosome. This means that males are not homozygous or heterozygous. They either have the trait or they don't. Remember, these traits or alleles will only be carried on the X chromosomes. So the men will have X big B Y, or they have X little b Y. There are many common X-linked traits, such as color blindness, male pattern baldness, and hemophilia. If we think about color blindness, which is a recessive sex-linked disorder, we can wonder, who does the man inherit that color-blind allele from? His mother or his father, or both? If we look at the genetics or genotypes of the parents, we see that the man only has one X chromosome to give. For him to have a son, he must give that Y chromosome to the son. The Y chromosome does not carry an allele for colorblindness. This means that if a man is colorblind, he must have received the colorblind allele from his mother. We're going to work through some X-linked or sex-linked genetics problems in the next slides. Remember, when you do sex-linked problems, that you're going to have to keep track of your sex chromosomes, the X's and Y's. You also need to remember that Y chromosomes are teeny tiny and they don't carry many traits. Therefore, they are naked. All of these traits are going to be carried on the X chromosome. So in fruit flies, we have eye color sex-linked, and red is dominant to white. So first step is to set up a key of alleles, just like any other problem. So big R is dominant, so red, little r is going to be white. Our male fly is white-eyed. This means that he has X, little r, Y. The female's genotype is homozygous dominant. So she's going to be X, big R, X, big R. So once we have our cross, we can plug this into a Punnett square. Remember to keep track of your X and Y chromosomes at all times. So dad is X little r, Y. Mom is X big R, X big R. We can plug this in. Even in our Punnett square, we're going to have all of these X's and Y's. So now we need to go back and answer what is asked. So this question was asking for the phenotypic ratio of the offspring. So remember that the gender is part of your phenotype. So in this case, we have two females, and they are both red-eyed. So 50% red-eyed females. In these other boxes down here, we have two males, and both of them are also red-eyed. So 50% red eye male. Be sure when you're doing these problems that you give me the full phenotype as well as the percentage or fraction. So hemophilia is a sex-linked disease that we see in humans. First, we set up a key of alleles. Our big H dominant is going to be a healthy individual. Our recessive, little h, is going to be the one with hemophilia. In the problem, it says a man who is normal, so no hemophilia. This means that he is X, big H, Y. And he's going to marry a woman who is a carrier. So remember, carrier is just another word for heterozygote. So it's referring to her genotype. So she's female, she's X, big H, X, little h. So remember to keep track of your X and Y chromosomes when you're putting these gametes on the Punnett square, and to keep track of them when you're filling in the Punnett square as well. So our first box is X big H, X big H. Second box, X big H, X little h. We have X big H, Y, X little h, Y. To answer the question, we need to report the phenotypic ratio of the children. So translate what is in each one of these boxes. So if we look at the top two boxes, we see that these are both females, and they have at least one copy of the big H allele, which means they are both normal, healthy individuals. So we have 50% females who are healthy. When we look at the bottom boxes, we see that there is one male with a copy of the big H, so he is healthy, so 25% male and healthy. The other box has a male with a little h. This means that we have 25% that are male with hemophilia. So the second part of this problem is asking about the chance of their son having hemophilia. So because we're just talking about their son, we're going to focus on the bottom two boxes. So we know that they have a son. So out of those two possibilities, one of those boxes has a child with hemophilia, which means that there's a 50% chance that their son will have hemophilia. So this would be a little bit different if I asked you what was the chance of a child having hemophilia. So the chance of any child having hemophilia is 25%. So at that point we're looking at the whole Punnett square again, not just the bottom two boxes.